Welcome, Howard. I don't think I need to say too much about Howard. As you know, he started this meeting in 1999, and I think it's for our final keynote. I think it's a pleasure that he's agreed to stick around to the end, and I think he'll be entertain us. This is terrible, so I'm just going to
Thank you, Aaron. It's a pleasure to be here, and I'm always stay to the end of this meeting, by the way. It's not true for other meetings, but I always stay to the end of this one. So it's really a pleasure to be here. I got here, and Joe said, well, that's a really cool title. When did you come up with that? And I said, what's my titles? And Joe thought that maybe I had been drinking when I came up with this title. So I don't think that's true. But what I want to do is try to put in a framework and give a lot of opinions. And the whole point is that maybe there'll be something in here that's useful for people to think about. And because Ann is in the audience, Kurt Sigmund, who is chairing our external advisory board for the R24, said the first slide that I have to have up is about that program. So Ann, you can go back and report to Kurt. It's not moving forward for me. It's the green, big green, that big green one? Oh, there we go. All right. So it is a resource for the community. It says the first round deadline was October 1. Many of you actually submitted to this. It's a very simple application process, and it's going to be, it was very fast. We just had the review last week, and I'll show you the applications that were in there. We funded 22. There's a little bit of space there, and we're going to do 50 gene modified every year for the next 4 years. So the idea is to make another 200 strains, and so we encourage you to come in and apply for this. The next application will be around March is what we're looking for. And they were supportive of foreign applications. So the keys are around heart, lung, and blood. So you have to be able to justify that and generalize utility for the community. That's what they voted on. There are other solutions around that. This is the list of the strains and the genes that have been agreed upon so far. So you have to also agree that the data will be available, the rats will be available, and you don't mind having this up here. Prioritization. If your application goes in, it will be done in the order that it's ranked by the committee. You will get the first breeder pairs that come out, and after that, it's whoever else wants breeder pairs. Now, we've had to redo some of this. There are some challenges around this, and we've dropped using ZFNs, and people have asked me about this. It has not been a very pleasant experience over the last few months. We had originally thought about using ZFNs because we've been using them for a long time, but Sigma sold off the IP to Sage completely, and it turned into a bit of a nightmare. So when we got the grant, I talked them down. Now remember, we got the grant. I talked them down from wanting to do every single injection for $80,000 a piece down to a third, which was still over 2x the cost what it would take for us to do that. So I thought that was at least okay. But then for pricing of the rats, which the reviewers were very concerned about, I was told that it's going to be $450 per rat, and there's no commitment what the price would be. And I also think that's untenable for a reagent that is developed by the community, for the community to help us go forward. And the last piece was also a bit strange, was they then wanted to make all of the animals, and then the license for distribution of the animals was the RRC. So we write the grant, they knock them out, and RRC distributes them. And so because of that, we've gone back to the NIH. We went through a very big series of discussions around this, and so we've been granted permission to actually drop using ZFNs. The EAB is actually quite comfortable with this, and so we have a completely different partnership. And it's disappointing, but that's the story. There's much more to this. I'm not going to browbeat anybody about this, but it just became untenable for us to do this. So we have a partnership with Transposogen, It is all underneath a patent license. It has no restriction on your animals. And there's not a problem anymore. So I think that solves a lot of problems. Aaron's been working with these guys for some time. We've done this with commercial entities as well. You can do whatever you want with the animals. It will distribute the animals, will make these available, and so I think it's just going to make it a lot easier. There are also some groups that want to do CRISPR, Cas9, and so we're going to look at that. But again, we're not going to do anything that ties up your IP or ties up our ability as a community to make these. The external advisory board is Kurt Sigmund, Joel Elmquist, Miriam Frenage, Gordon Mitchell, and Marcelo Nobrega. I told you what the relevancy around this, and the program officer is Deborah Applebaum-Bowden. It's a great group. They were very supportive of people's applications, and they're very interested in trying to help you guys get your animals through. So it's a nice problem for us in that we are going to be able to help you get your animals done, and then that makes it a lot easier. Okay, so end of that. So you can report back to Kurt, please, Ann, that I did exactly what he told me to do. This is the group of people that have been in the lab for some period of time. It's a group of investigators. It's Aaron, it's Mindy, it's Joe, it's Carol. So it's a bunch of different labs and a bunch of great students and postdocs and technicians who have done some wonderful work that I'm going to talk about. I'm going to talk a little bit about our clinical work as well. I think the important thing about this is that this is all of our clinical program. There's only 4 physicians on here. Everybody in the middle are computer people, which kind of tells you where all this is going. So with that, I'm going to give you a little overview. I heard this over and over and over again, and I have the same feeling. We have a full genome toolbox, and it seemed like nobody likes the rat. That's not anything new. Those of us that have been doing this for a long time, we have to kind of accept that. So I don't think we should be discouraged around that. And I'm going to talk around why I think we should be excited about this. People have started doing this. We've started doing this as well, is genome GWAS to function and leveraging the model resources. But I think we can take this much, much further, and I'll talk some more about that. The exome sequencing and whole genome sequencing is going to drive even more data around this, and I'm going to really spend a fair bit of time on that. And suddenly, I think it gives us some unique advantages. But the key has to be, in my opinion, is we've got to be attacking human variation. That's got to be what the strategy that we're going to go after. What makes us different? What makes our models important? What makes these technologies valuable? We've got to build better models. We've got to be reflective on that, and we've got to understand what will people fund and what won't they fund. And we need rapid resources. It's not sufficient for some of these projects to wait 4 years to get something. And these pieces now become incredibly important. And as a community, we have a huge volume of activity that we can share, and we do a pretty good job between each other to do that. So all these strains have been sequenced. That's made a fundamental shift in how we view the world. We can now go in and take a look at this data. We can get all these pieces. We can have all this variation. We can go down to individual gene variants, and we now feel like our QTLs and everything that we've been working on for 20 years in some of our cases should just be obvious. It should just be obvious for funding. But the world's changed, and the driver around this, in my opinion, is now the human is the better model for finding variation. But what do you do with the variation? And what's happening is people that are doing this are discovering the haplotype blocks are quite large in human. And you have all of these variations inside that haplotype block. What do you do with that? So we have a huge resource that's sitting around. We've got 256 strains on the hoof. We've generated almost 140,000 litters over the years, and this is all available if people are interested in this. Let us know if you need the strains. Let us know if you have a need for some of this. It's all there that's available for that. We have Akiko who's been doing amazing work for us. She did amazing work with Tadao and Berger et al. in Japan. She's got magic hands for being able to do everything from ICSI through everything else that's available. So if we can help in some of these places, if we have animals that are of value to you, let us know. Because what's starting to happen is we're getting contacted by people. Hey, do you have a Rat 4? What about this? Can you do this quickly? How fast can you do that? And so if you're getting those approaches and the rats are available, then we'd be more than happy to share those with you. So my opinion 1, human GWAS to drive your research. We've started hearing some of that. There's quite a few great talks on that from multiple groups. And we've got all of this data that's sitting there. So here's a bunch of QTL plots. This was a lot of QTLs generated by John Rapp and his team. And you can now find where these overlap. You can see where all these things live across the human genome, across the rat genome. And then you can take the GWAS data and overlay it. Use the GWAS data as a way to help prioritize your genes that are inside that. Now, in my opinion, and this is what we do in our group, we're only going after QTL now for which there's GWAS targets inside there. It doesn't mean the other stuff isn't important. We just think it's easier to sell. I don't have to justify why is that important. There's a GWAS marker there. If you're funded to do this and you have the resources to do the others, then that's great. But think of that as a way of going forward on this. And so when the GWAS studies came out, there was a whole bunch of information on this that none of us knew what these genes did. We were surprised about these different bits. You heard about a bunch of these genes. Brad Enders gave a great talk on pleca7. So there's these strategies that we can now go in and attack. So there are a bunch of targets that have already been made. They are available, 100 GWAS targets. If you're interested in those, let us know. And like, for example, I was talking with Martin about, UMOD, and Martin thinks that it died, so we need to find that out. But if it didn't die, Martin now has a rat that's available, and if it did die, well, then we don't have a rat available. And you can go to RGD, and you can go and click on these, and it tells you where all of these different genes are. Again, if you're using a QTL, and it maps inside a place where there's been a gene that's already been targeted, maybe it matches your phenotype. Maybe it doesn't. But I'm just going to argue that this is a way to potentially accelerate and drive this forward. It's too bad Ted had to leave because I just was going to have to poke him a little bit, that this is a circuit diagram, kind of Guytonian, if you will, from a project that Mike Flister just had published in Genome Research. And in this case, here's another example of what I think you can use a rat for in order to address a complex question you could never do in human. So this region, AGT trap plod 1, has been associated in all these GWAS studies. But there's multiple genes inside this haplotype block. So which one is it? We make the assumption, and the authors make the assumption that a gene is responsible, but maybe it's more than one gene. So with Aaron and Mike and the team, we went in and we knocked out all of these different genes in there. And what did we find? Well, we found that there's multiple genes having an impact. Some are causing an increase in blood pressure. Some are causing a decrease in blood pressure. But they're in the same haplotype block. And not only that, we found that when we took a look for renal disease, we saw the same thing. Some caused an increase in proteinuria, and some caused a decrease in proteinuria. There is no way in a clinical study you can attack that. There is not the power to be able to do that. So something to think about this is that if you go back, some of you are way too young to remember this, but cystic fibrosis. Once people knew what the cystic fibrosis gene was, then a variant inside that gene then became a potential pathogenic marker, okay? So these genes now have potential pathologic markers in them, and now Stuart Cook and others that have their human samples can go in and say, okay, are there some variants that can account for that? How do we model that? So you need to have that type of information. Because inside this one locus, there's multiple alleles. Some increasing, some decreasing, and some keeping it the same. And so we believe that looking at that is another option and another way that as investigators we can have an advantage and we can leverage this type of data. And I don't think this is going to be unique. If you look at all the congenic data, John Rapp stuff over the years, he kept chopping them up. And there's multiple phenotypes inside those regions. That means there's probably multiple genes that are in there. Opinion 2, humanize your rat. Well, there's a bunch of ways to humanize your rat. You can obviously put human genes in. You can do transcripts. You can do all those things. You have to pay attention to, do you have the right machinery that's involved in all of that? But you can also do what Mike Flister and Aaron came up with, which is a clever idea of You can also humanize by doing transplantation studies. So make a skid rat. Sorry, I said mouse. And you then end up having the ability then to do some transplant studies. So they've done a clever strategy of taking human cancer and putting it into the rat. But there's all kinds of other things you could do around that. And in this particular model, they're able to show that these different backgrounds have a different effect. So again, it's a rat model that we've always done something with. We've been studying breast cancer in this community for a long time. It's great stuff, but we're just turning the page a little bit, in my opinion, to be able to do that. And so you now have this ability to look at the different changes in these and go in and ask questions that, again, would be very difficult to do inside a human. So how do we take what we have and turn it into something that's important? The other part of this that I think, and fundamentally this is where we're going, at least my group is going, I'm not speaking for Aaron and Mindy, but for my group, we're really moving into a completely different way of thinking about this. And it's really talking about how do we take variation and deal with variation for a patient. And so I'm going to talk a little bit about that. So some of you know this story, some of you don't know this story. So I'm going to start with, this is a little boy, Nick Volker. He's 2 years old at this point. Happy, normal little boy. At 4 years of age, I got an email from a pediatrician, a gastroenterologist for this kid. And I can tell you now, Nick is a 4-year-old boy with a very unusual malignant form of inflammatory bowel-like disease and profoundly delayed wound healing. He first presented with weight loss and perianal abscesses and was on a downward spiral for over 2 years. He was admitted in January with a life-threatening breakdown of his intestines. He was in the operating suite every other day. He lost his colon, and no one knew what was wrong with him. And Alan said, "Can you read his genome?" We weren't sure we could do it. We thought about this and said, okay, let's try it. It cost $75,000 in 2009 to read his exome. So can you find something with that? So we set out to do that. This is what Nick looked like at 4. So he was in much worse shape, and he was on the verge of dying. And so what we discovered is sure enough, you can do this. You come up with about 16,000 variations. And Liz, who was a research scientist with RGD, worked with some of the RGD guys to actually build a software program to figure out how to analyze the data. How do you find one variant? Because there was nobody else out there. And how do you prove that? How do you make those decisions? So I'm not going to spend a lot of time on this particular case. I'll talk about another case and illustrate some of those points. Long story short, we found a novel mutation that had never been seen before in a gene that had been seen before, XIAP. He got a cord blood transplant, and Nick is 9 years old now. He's doing very, very well. So that was a cascade of events that happened around this. It got a lot of attention from a lot of different people, but the most important thing is that it helped this individual kid, and it really got our group starting to think about, can we do this in more and more examples? So this was the paper. We had a very difficult time publishing this. This was the 7th journal that we sent this to, and we basically had a tough time. So here we save a kid's life by using genome sequencing, and it was questioned because it was an N of 1. Well, guess what? When we sequence the whole world, we're all going to be an N of 1. How are we going to do this? There's not enough power if we sequence the entire planet to solve a clinical trial for every variant. So how are we going to do this? How are we going to attack those different types of pieces? And this is something that we're starting to spend a fair bit of time on. How do we prove a variant quickly? So we have built a complete clinical program. We do whole genome sequencing. We do exome sequencing. We have 3 geneticists. This is what they do, 2 genetic counselors that are involved in this program. And it's been for rare diseases for the most part. But we're now thinking about how do you expand this out into other different areas. Well, we just published this, the first pilot project back in July, and we talk about the different types of cases that we've looked at. And what we've discovered in looking at this is that there's a very difficult series of data sets. So I'm going to spend like 2 minutes on this, but we have variants. We have pathogenic and these things called a VUS. So these are variants of uncertain significance. And you can see, well, we have these 4 pathogenic, but we didn't make a diagnosis. We have 3 pathogenics, and we have 11 variants of uncertain significance, and we maybe have a diagnosis. Versus down here, we have 1 pathogenic and 6 VUSs, and we now make a diagnosis. So how do you reconcile the pathogenicity of variants that are known with the disease? How do you do this when there are no other patients in many cases to be able to do this, right? So this is really fundamental individualized medicine. So how do you make those decisions, and are you going to change care based on this information? The other thing we found is that we've now landed a couple more of these. Several of the maybes have now been resolved. And what we also know is we're making 25% more diagnoses by doing whole genome sequencing than doing exome sequencing. So there's more information that's there. So how do you take this information and press forward on this? Well, it's not just the genome. There's a whole bunch of other things you have to piece into this. How do you take all of this information and glue it together and turn it into something? And again, the advantage of having had the Rat Genome Database and having people like Mary Shimayama and Liz Worthy, we could then build a tool that then takes this information. So how do you take all this information out of the electronic medical records and turn it into something that's usable? So here's another case. This is a case for Andrew. We call him case 48. And he had 10 years of data that was collected. He had all kinds of disease syndromes that he was tested for. He had all kinds of medications that he was giving over the years. He had a series of strokes, and there was debate among the care team about whether or not it was the drugs. All these laboratory tests. When did it happen? When did it start? What time? How do you divide this out? Which is the beginning piece of this, right? So the great thing about this community, and we were talking in the bar last night about this, is that the rat labs are moving into this. Tim, Edwin, Stuart, Norbert's group are all doing this. We don't know any mouse lab sequencing group that's moved into this, right? So we have a way of thinking about this that's quite different than other people. And I think that we can leverage that type of information. So this case turned out, we failed on this case, but the NIH, the UDP program, actually was able to solve it because they had additional cases around this, and they could then go in and find the cause of this. And it turns out that the drugs weren't causing the strokes. It is actually a phenotype. And they proved it. It's in the process of being reviewed. I don't know if it's been accepted yet. And they went in the process of using zebrafish as a way to prove that there was leakage in this that caused this. And the reason we didn't find it in Andrew was because there's a deletion in a repetitive area that the small sequencing for next generation sequencing missed because of assembly. The only reason they found it was because they had a different gene variant in their family that had the same phenotype. So they spent a lot of time working on it. So the lesson in this is, how do you take this information and move it forward? How do you pull these different combinations together? We had a great discussion in the bar last night around this, so I'm going to put this out there. So Stuart Cook and I think we should sequence babies the day they're born. Tim and Edwin think that's just atrocious. And we had some great debate about this. Irresponsible might have been the right word. So what's the right word? How do we do this? What's the right way to do this? And you're each going to have some opinions around this, but how do we think about being able to do that? And a question I get all the time is, why test if we can't do anything about it? And I annoy my clinical colleagues every time I do this, so we have a couple MDs in the room, so I'm going to apologize. It's intentional, okay? So I'm going to argue that we do this clinically every single day. We give diagnoses every day for which we can do very little about. Hypertension. Stuart and I had a great conversation about at lunch the other day. So is it really that we have bad drugs, or do we have poor compliance and we don't practice medicine the way we should? How about cancer, diabetes, obesity? How are we with those diagnoses and curing them? I'm going to say we don't do very well. And so genetics is really no difference. There's all kinds of things that we can find. So here's another case. I don't think many of you in this room have heard this story before. My group has heard this about 12,000 times, so I'll apologize for that. But this is Lillian. So Lillian we saw her at 4 years of age. She was a 30-week preemie. She had, the mom had a lot of pregnancy complications, so you now start wondering, is this environmental or is this genetic? Following an emergent C-section, Lillian had a whole bunch of different problems that were associated with this. They were trying to figure out what was wrong. She was in the NICU for 3 months. Nothing was ever resolved. There was tons and tons of tests that went on, and extensive evaluations, extensive work went on, and ultimately she ended up in Children's Hospital 4 years later, and the decision was made to sequence her. So we sequenced her, and we found a gene that we think is potentially causing it. It's called SCIV2L. Well, this is the entire list of gene information around this. 13 papers, none of which were related to the clinical phenotype, and the clinical team was pretty underwhelmed by characterization of sheep as being evidence for something that we would do for this case. So what do you do with this, right? So in this particular case, we had done a functional review, and this gene is a binding partner with TTC37, which is actually a syndrome that ends up causing very, very much the same phenotypes that Lillian had. So we found a variation in SCIV2L that plays a role with this, and it's a binding partner. So would you make a diagnosis with that? Now, most of us, as researchers, don't know what to do with that type of a question because we don't know what the criteria are. But the decision by the clinical team was there's not enough evidence. What's the clinical evidence? And she wasn't in severe case like Nick was. Nick was dying, so we had a little bit more latitude. But Lillian wasn't dying. She wasn't doing well, but she wasn't dying. So could you make a diagnosis on this? So we actually had this variation for about 4 months before this paper came out. And the paper came out that identified 6 individuals with a mutation. And it turns out Lillian has exactly the same variant. So we were now able to make a diagnosis, but what did we find? Well, we found there's no cure, there's no specific guidance on treatment. We're not sure what to do. The family wants to have more kids. They can think about this. And so in some cases, when we present that, people say, well, you shouldn't be doing it. This is really irresponsible, right? Because you can't do something about it. So what value is it to be able to do this? And there's something that we're learning as we do this, is there's something called personal utility. And what we're finding in genomics, particularly for these very rare diseases and pediatric cases in particular, human nature needs to know answers. And we're finding even in negative cases, the family knows 2 things, and this is quotes that they've said to us. We now know it's not our fault, and we at least can stop looking. So I want to do a video and show you what families say about this, okay? It's a little dramatic because it's designed for raising money, so I'll apologize for that part of it. But I want you to listen to the parents, and I want you to think about this as we go through.
Our whole life, since Lillian's been born, has been kind of like a roller coaster ride. She was born at 30 weeks, 1 pound, 4 ounces, 11 3/4 inches long. She was ill several times in the NICU. She was tested for Shwachman-Diamond syndrome. She's been tested for cystic fibrosis. She slowly stopped eating anything by mouth, and we just did tube feedings. She's had 3 colonoscopy endoscopies. I can't even remember all of the different things she's been tested for.
Many institutions. They had been seen by many doctors. They'd been seen by geneticists. They'd been seen by all sorts of other subspecialists.
She had been through so many different tests, and with them all coming back negative, I think that at times that they wondered if they ever would find a diagnosis.
She became sick in fall of 2010. I remember looking at her one day and feeling like she was dying in front of me. She had gone from 24 pounds to 18, and they couldn't figure out what was wrong. So that's when we came to Children's.
You know, the whole concept of the whole genome sequencing was new to us.
Genome sequencing is a process where we examine or we look at all of the genetic material, all of the DNA, all of the instructions that a person has.
Its purpose is to help us diagnose children when we've not been able to find a diagnosis before. Many physicians suspected she might have a genetic condition.
She really fit many of the criteria of a patient who had an unexplained disorder and for which there may be potential treatment strategies.
Genomics is almost our fulcrum, if you will, to the reach out to the many, many specialties that our genetics program supports.
One of the remarkable aspects of whole genome sequencing is really the range of patients that we've been able to help. Patients with intestinal problems, patients with immune problems, patients with neurologic problems, patients with cancer. The list goes on and on.
If they could find something that would tell us why Lillian is who she is and why she has the problems that she has, that was our number one thing.
Anything to give her a better life.
When we finally found the molecular diagnosis for Lillian through her genomic sequencing, we were just ecstatic. I couldn't wait to pick up the phone. I couldn't wait to call them.
She's the first child that's been diagnosed with this in the United States. She's unique, the diagnosis is unique. We have an answer. It gives something for the specialist to continue to look into, to see what they can do to better help us.
Lillian has made tremendous progress over the years. Every time I see her, she's doing better and better.
By doing the whole genome sequencing, we don't have to keep searching. If it weren't for Children's Hospital, we're not sure if Lillian would still be here with us today.
So the question is, when do you deploy this, right? When do you choose to do this? You know, is this good medicine? Is this actionable? So we would argue it is actionable. There are groups that actually don't agree with us. They don't think that's actionable at all because we can't offer a treatment around that. And I think these are just areas that we have to face. But when you're thinking about what you're doing in your lab, we don't tend to think about it this way. We think about we're trying to understand disease, but this is real. We're trying to impact people, and we're trying to figure out how to go forward. So this is one of my nightmares, right? We were incredibly lucky. This variant we knew for months. What if that paper hadn't come out? We still would not have a diagnosis for her, even though we were right. How do we do this, right? And I'm going to argue what you all are doing is one way to do that. But we have to be faster about it. We have to be smarter about it. We have to think about how do we test this. We're now pushing our clinical teams to make diagnoses on functional studies. What will they do it for? What's the criteria? How do you set that up? Really uncharted territory for a lot of these things because we can't do a clinical trial. There's 6 kids in the world. How do you do this? So we've started working on another program of how do you attack this, right? And so we're looking at a different series of opportunities. So we're looking at white blood cells if we can. Sometimes you can do the phenotype there. We're doing inducible pluripotent stem cells with Steve Duncan and being able to test there. We've started really looking at zebrafish, and we've starting to look at cell functional assays and other examples of being able to try to deduce these down into a much more rapid protocol. So with Steve Duncan, we now have the ability to take these gene editing technologies, and we're actually working with Sangamo on some of these because they've done some clinical trial work on some of these. So you can actually now go forward and working with the different stem cells. And this is work that Kareem has done in Steve Duncan's lab. And what they're able to do now is be able to go after in liver, and they're able to build these human iPS cells from a patient. Now the problem with this is it's just slow. It takes about 9 months to get to a point, so they can do it. We can get to a point where these things really do look like they're behaving like a liver, right? And this is for another case that they were taking a look at for MODY1. So you can now go in and show that it has albumin, and it can make lipids, and it can do all the other pieces around that. Histologically, it functions like a liver. You can show differences between the adult liver and the fetal liver. But will you make a diagnosis with this information? That's the question, right? That's the challenge around this. Now, of course, it's going to come down to what's the risk-benefit ratio, right? If the patient is really, really sick, you're going to have a different level of flexibility around that than if they're not. So that just shows you can get to the pathological point. Well, Flo showed a study on, that we've been doing for zebrafish. And I told you that the UDP program, the Undiagnosed Disease Program at NIH, is doing this. And so we're also doing this, where we're now taking, instead of looking at the rat example, like what Flo showed, is now being able to go in and modify genes in a way to attack specific VUSs, and now look to see if we can mimic these. So again, you can use the gene editing technology to go in and start addressing those questions. We also have another case where we have found the gene for a family that has a very rare form of kidney disease. Okay, it's been published, but no one knows how this works. So we're now in the process of making a rat where we're modifying that particular variant, and we're now going to test and see if that works. But in this case, we've got the advantage that although they do get renal disease, they get it late, and it doesn't kill them right away. So we have time. But how do we do this in cases where we have a kid in the NICU? How do we attack this type of information? So the conclusions, hopefully I've tried to convince you, is that we as a community have an amazing wealth of resources. We've got animal models, we've got gene editing, we've got different expertise, and as a community, we get along pretty darn well. And I've noticed some things happening, right? So what tends to happen when times get tough, people retract into themselves and people get aggressive. And I'm going to argue that when times are tough, it's when you have to come together and you have to work together. That's how you get through the tough times. And we've got a tremendous amount of opportunities. We have a large number of strains, so do many of you, right? How do we use these technologies to help advance each other? That's the trick. That's what this community has done very, very well. Which of the young people in the room are going to be the advocates? Who's going to drive the community in the future? Who's going to make these things happen? These are choices that have to be made. And we're all complaining about grant reviews and paper reviews, okay? We review each other's in many cases. Are we doing the best for science and the community when we review? Or are we just being negative and aggressive? These are questions we have to ask. These are important issues we have to face in these kinds of times. The special part of this meeting has always been that this group has worked together as a community. Let's keep that going. There's huge opportunities here. This genome sequencing stuff in the clinic, it's going to get faster and faster and more, and there's going to be all of this data that no one knows what to do with. I think it's an incredible opportunity to help patients, to do good research, and to advance knowledge. But it's not how we set out to do this. We set out to do QTL hunting. We set out to find our genes and then to take those to human. But people like Stuart that have these huge cohorts, right, they're finding variants. How do we help Stuart answer those questions and other physicians around us? How do we do that, right? That's what this is about, in my opinion. I also think that gene editing is a powerful tool. It's not just for research. It's also for clinical. So Sangamo has 2 clinical trials that are going on around this. We're going to see this happen more and more. Well, where should we test that? I'm going to argue the rat. The cool models many of you have, those are potential models for being able to do gene editing. How do we target kidney? How do we target heart? How do we do that, right? Fascinating opportunities for us as a community. So this is a debate. I'm going to go back and I'm going to finish on the debate that we were having last night in the bar around this. And Tim and I are never going to agree on this. And that's okay. I mean, I think this discussion is really important, and I'm putting this out there not as a statement about Tim's right or I'm right or whatever, but I think these are important things that we as scientists have to participate in this debate. So I think we should be sequencing at birth. And this is the major case why I think that, right? Now you can argue how many failures could we possibly have happen to get to one success. But this is one of our kids who initially presented with poor feeding and failure to thrive, met early development milestones, and then began to fail to meet the milestones and develop seizures. Many, many tests were performed. We didn't know what it was. Her presentation phenotypically wasn't anything that was obvious, so we had done everything that we knew to do. 3.5 years later, the phenotype actually evolved to a point where they realized that she actually had a vitamin B12 disorder. So she was started on treatment, that's the good news, and the neurological abnormalities stopped. But she's permanently going to have neurological defects. Now, no one made a mistake. The phenotype wasn't typical. It wasn't what was expected. Had we sequenced her at birth, we would have known this. Now, what's the cost benefit to that? I don't know the answer to that. I really don't. And in this case, I agree with Tim. We don't know. We have to be careful with this. We have to be responsible around this. But fundamentally, it drives me crazy that we've missed some of these, right? And so our philosophy is we don't want to have any more sick Averys. Now, how do we do it ethically, responsibly? I don't know. I mean, I think there's a lot of questions around that. So with that, I'm going to close and take any questions. We're in an amazing time. It's tough. But you've got cool stuff. You've got great opportunity. Let's leverage it. Thank you for your attention. My good friend, Dr.
So Howard, that was a great talk, an inspiring talk, and I think the balance between rat and human is really exciting for this community. I just wanted to make a few comments. It is a strong community. It's a remarkable community, this group of people who are in this room and have been in this room for the past 3 or 4 days. And I think we do get on very well with each other, which is not the same as other model organism communities necessarily, and certainly the human genetics community, but it will be competitive and it will continue to do so. And I think at these times we should stick together. I think what is clear from the presentations and from what you've said is that there is excellent science that is being done, and I don't think we should compromise on that. It's the excellent science that will continue the funding and that will continue to enable us to do the things that we enjoy doing, and I think all of us should continue to strive for that. I think just on the point about the sick children, you'll get the last word on this, so that's okay. I mean, I think I would just point out that there is a difference between sequencing children who are sick or who come with symptoms as opposed to sequencing healthy children, and I think that that is a debate that I'm sure we'll continue to have. We're probably not, well, we probably are a way apart from that, and it may take time before we come together, but I think sequencing sick children and sequencing healthy children are two different things. But in terms of what is true for the rat community, there will be fashions in funding and economic cycles, and model organisms don't seem to be favoured, particularly in the EU, and you've done a great job for raising funds for rat knockouts and things like that. I think I come away from this meeting, I came here because I've been coming for ages and I was asked to come and speak, but actually I come away inspired with some of the science that's been done here. I think the science is excellent, and in fact it will encourage me to stay with some of the rat stuff that I've been doing for years, and I will definitely continue it alongside the human stuff, but the rat stuff, I think we shouldn't just be persuaded by fashions that, you know, say, oh, model organisms are not the thing and translation is the essential thing. I think we should stick with what we've got. There are some great stories. There are some, even if it's not translating human stuff back to the rat, there are some great rat stories that need to be taken forward, and they will be taken forward if we stick to scientific excellence. So I feel very inspired, and I think that I will try and do the same for the people in my group and elsewhere who are going to continue to work with the rat.
First, when you talk about community, I'm pleased that I have had a chance to spend 3 days with this community. It was a wonderful experience. the issue of genetics and sequencing, philosophically, has to be taken in the context that penetrance and environment are still important. And that's another area that we have to solve. That, you know, if we were only dealing with Huntington's, fine. We can say that in 40 years, the person is going to be affected. But many of the, and particularly in the area that you are with hypertension and things like this, there are lots of variants that need to be brought forth. And when people ask, what can you do? I think that's one of the things that is very important to let them know, that you maybe don't have to engineer the gene, but there may be other behavioral or environmental approaches that you can deal with some of these variants and still get very good outcomes. So I think we should also not forget that side, that we don't have to treat genomics with gene therapy, but there are other alternatives as well. And really nice. Thank you.
Thanks.
Howard, I thought that was really inspiring, and I want to thank you for giving us an optimistic view, and I won't go into the litany of grants that you know I've not gotten because the reviewer can barely consider that I'm working in the Rat, and writes, we really don't know why she doesn't do this in the Nod mouse, literally as stated in my review. So I think I just had that one comment that you say, are we doing the community a service by when we review papers and grants, or are we just being picky? I can tell you they're being picky. They're being damn picky right now. And unless we work with the NIH to see that the bigger picture, they're going to go to a model where they just fund important friends of NIH with DP, with directors program projects based on the reputation of the person and not necessarily on the quality of the science. And so I think that we need to volunteer to be on study sections. I think we need to not turn down papers because we're busy, to review them so that we as a community and people who are aware of the cool science going on here can struggle for it and fight for it in those venues. If we say no, we're too busy, then you're going to see, well, in the diabetes field, you're going to see the perpetuation of science that's carried us nowhere toward a cure of type 1 diabetes. And so I support what you're saying, which is largely important for single gene defects that can be detected by sequencing. But we really do have to shake up CSR and make sure that the rat is considered a viable organism for study. I don't know how to do that.
If there's no more, oh, sorry, John, go ahead.
I have one question. Well, actually, a question and a comment, I guess. As part of the rat community, I think it's very interesting in that we all work on very different physiological aspects. You work on heart, lung, blood. I work on cancer. But the genes that you're finding are not just heart, lung, blood. They're also related to cancer. And so I think we need to think about that not only from our perspective here at a meeting, but also between the NIH subgroups. So I think about things like the neuroscience talks that we heard this morning. The number one place that we make serotonin is not in the brain, it's in the gut. And it's related to things that we're not thinking about. And it's those interactions between organs and systems that we need to start thinking about in the rat, which is, I think, much more easy to draw that interaction than in the mouse. And so on a practical note, are things like the tailends and things that you're generating, even though you're generating them on the backgrounds that you're interested in, can we use those? Are those resources going to be available to people to do in other strains?
Yes.
Good.
And we'll be happy to make other strains for people also, right? I mean, I can't control what heart, lung, and blood wants to do, but I will tell you that based on just what you said, most genes, and Mary and the RGD guys would be more than happy to help you find your cancer gene's relevancy to heart, lung, and blood, right? So there's nothing wrong with putting that in there and then being able to say, hey, you know what? It also benefits cancer, right? So if you're looking at the ability to expand this into more places, that's perfect, right? Because then it's a twofer or a threefer, depending on what these genes are. And that's why I encourage people to go look at what's there, right? It might have already been available, or it might be available, or it may be coming available. So I agree with you 100%. So, and if it doesn't work in that, then I think we can also talk with the NIH groups and say, hey, if we're getting a bunch for addiction, right? It doesn't cost the addiction NIH branch very much money to actually just add that in as another dimension. And so again, I think it's about, you all have to support that kind of an activity, right? If you guys want something like that, we're more than happy to do it. I mean, I did this grant because I felt that we needed one more round of large numbers of these things coming out so we could try to break the barrier where people don't accept the rat, they don't accept those types of different things. So the more we do, the better we all are, because the better the science, the greater the publications, then the easier it is for us to get grants down the road. So absolutely, I agree with you 100%.
So Howard, I thought that was a fantastic talk, and it gave us all a lot to think about. I just, one minor comment, I wanted to clarify something you brought up at the beginning about the RRC and the agreement that we have with Sage, and just to let people know who are making knockouts or anything modified with zinc fingers, is that agreement is not restrictive. That was part of the reason we tried to get an agreement with them, is so that you all could deposit your models in the RRC and they could be distributed freely without restrictions.
Okay, I would like to contribute a little bit in addition to what Howard's doing, in the sense that I think it's a really, really inspiring discussion, and I appreciate that Howard actually put his talk in the end as an anchor, which is a wonderful thing that he did. Now, I believe NIH, I'm not talking officially for any other institute or anything, but on the other hand, apparently NIH justifies the funding according to the necessity and uniqueness in rat. So I believe we have this, everybody has this particular responsibility for dissemination. We try to tell everybody why rat is necessary, and we try to tell why genetics is the future for many other research. And if people are convinced, understand, then there's a much easier way to actually get rat funded for many of the things. And one of the other part I can also maybe contribute a little bit more is that I had some discussion with some pharmacological drug development company, and what they say is currently, because genetics and genomics is done mostly in rat, what they need to do in developing drugs, two translations. One is translating into rats, because all the drugs are tested done in rats, and then translate into human. And so maybe one of the dissemination of scientific field is that the rat is a pharmacologically much viable viable model to use, and I think that's a valid point to emphasize in many of the situations. Okay. Yeah, thank you.
So I want to close by thanking the organizers, Aaron and Michael and Bina, and I forgot one. Edwin. Sorry, Edwin. It's bar time, I'm sorry. I had a chance to do this for many years. It's a lot of work. And you guys put on a great meeting, and so I just want to close by saying thank you to you, because we all get to be here because you guys pulled us together. So thanks.